The goal of my laboratory is to bridge the gap between basic science and clinical practice. And I have already translated several of my discoveries. But today, I'm going to talk about our most challenging, and I think it's a high-risk, high-reward type project over the coming 25 minutes or so. So I hope you will understand the concept behind and some of the data I'm going to show you where we are at right now. So let me begin. So I think all of you know that 2 years ago, hypoimmunogenic pig heart was transplanted to a patient, and he survived for 2 months. I think this was a very significant achievement in the history of transplantation medicine. And also, another trial was done using pig kidney xenotransplantation, and to the, interestingly, brain death patient. And the graft stayed without hyperacute rejection. So these are what we call xenotransplantation. We're trying to use pig organs, but after genome editing to reduce the immunogenicity. This is fine, but I think even after successful transplantation, I think patients must take immunosuppressive drugs, as you all know, and this has, of course, issues for the quality of life for the patient. So what we are trying to do is shown here. This is our approach. We are trying to generate transplantable organs using the environment of developing pig embryos. So it's an in vivo generation of human organs. Okay. So the stem cells that we're going to use is, as you know, induced pluripotent stem cells. So this is a really novel technology. So we can reprogram, we can rejuvenate mature somatic cells like skin cells, blood cells, into a stem cell, which is functionally equivalent to ES cells. But unlike ES cells, we don't need to sacrifice embryos, so there'll be little ethical issues. And also, as long as we use ES cells, you really cannot get your own cells, tissues, organs. But with the iPS technology, we can make your own iPS cells, a patient's own iPS cells, and all these cells, tissues, organs are patients themselves. So this way, if we use this stem cells, we should be able to eliminate immunological issues. So the idea is kind of crazy, I know. So suppose this is our ultimate goal. Suppose this is a patient with end stage, say, heart failure. So we can put him on artificial heart. But meanwhile, we can generate iPS cells from this patient and inject these cells into a embryo of, say, a pig. But this embryo has been genetically modified so that they cannot form a heart. So if we inject these patients, the iPS cells, into these embryos and make a chimera, mixture of his cells and pig cells, then we should have a pig with heart. But heart should be composed of only of patients' iPSC-derived cells, because host pigs, they cannot form a heart. So this was the idea. So once we could obtain this chimeric pig, we can take out this heart and transplant back to the patient. Although this heart was formed in the body of pig, the cells are all from iPSC, patient's own stem cell derived. So this was the idea. So I'm sure you think that this is a science fiction story, but I think we have sufficient proof of concept data using rodents. So again, I'd like to show you the concept behind. We postulated organ niche, space for an organ. So in a usual chimera formation, we just mix normal animals' embryo and pluripotent stem cells, and then we can make a chimera. And because both of these cells can differentiate into any type of our body, so what we get is the chimera, where we can find cells derived from both normal host and donor cells. So this is general systemic chimera. But what we're trying to do is, we call it grass-cyst complementation. So instead of wild type animals, we use organ deficient animal. So if we mix these two and make a chimera, then if this donor has the target organ, so that we can, so this animal cannot form, then only pluripotent stem cell derived cells can make this target organ. So, so pluripotent stem cell derived cells developmentally compensate the defect in the host animal has. So this is the principle, and this is the idea of organ is to, of course, it's a conceptual idea, but this is the idea that I had years ago. So, I have to show some evidence for this. So in this case, we used a mouse genetically modified, edited, so that they don't have SAL1 gene. The gene is knocked out, and so this gene is important for formation of kidneys. So this is a photo of a newborn SAL1 knockout mouse. They don't have kidneys. They have ureters and bladder, but just they cannot form kidneys. But when we transplanted, injected wild type normal stem cells into this SAL1 knockout embryo, then we are able to see formation of kidneys, and they're connected to the ureter, and the bladder is full of urine, indicating these kidneys are functional. And under the fluorescent light, as you can see, because this is a systemic chimera, we see some GFP signal. But if you look at kidneys, they're very bright, indicating that these kidneys are mostly composed of injected pluripotent stem cell derived cells. So this was our first proof of concept data for stem cell derived organ generation. But these are mouse, so we cannot make human embryos without kidneys. So eventually we have to do this across species. So as a model, we used mouse and rat because it's easier for us. But they look very similar, but they are different species. They cannot mate, interbreed, or they have different chromosome numbers. And also the rat is at least 10 times larger than mouse. So using these two species, we generated first rat ES cells, rat iPS cells, and try to make chimera between mouse and rat. As you can see, even neonates, you see the difference in their sizes. So we next try to do this. That is generation of mouse with rat pancreas. So we try to show that we can generate stem cell derived organs in different species. So in this case, we used PDX1 knockout mouse. PDX1 is an important transcription factor for the formation of pancreas. So the target organ is pancreas in this case. And we injected rat pluripotent stem cells. And luckily, we were able to generate chimeras. But interestingly, the size of the chimera was a mouse size. We are expecting to see 50% black, 50% white, medium-sized chimeras. But that was not the case. The chimeras were small, mouse-sized, and although we were able to see formation of pancreas, all rat stem cell derived, but they are mouse-sized. So this mouse-sized pancreas was too small to transplant back to rat. So we performed the opposite experiment. That is, now we generated PDX and knockout apancreatic rat, and we injected mouse iPS cells. And this time, as you can imagine, we got rat-sized chimera, and we're able to see generation of pancreas made of mouse cells. But this time, this pancreas was rat-sized, and huge pancreas we obtained. So this is the size of the pancreas, mouse pancreas generated in rat body. And this is the control. So you can see how big this mouse pancreas is. So this was too big to transplant back to mouse. So instead, we tried to perform islet cell transplant, islet transplantation. So we prepared islets from this gigantic pancreas and transplant back to drug-induced diabetic mouse to see if these islets are functional and to see if they elicit immune reactions. They shouldn't, but just to prove it. So before transplantation of islets, we just analyzed. This is the islets from the knockout host, and these are the islets from the wild type. As you can see, with the knockouts, because of the organ niche, almost all of these cells are GFP positive, whereas in the wild type, it's a mixture of both cell types. So anyway, these cells, these islets, look very homogeneous for GFP positivity. But when we analyzed using the FACS, we were a little bit surprised to see about 5% of cells are rat origin. I think these are endothelial cells, like CD31 positive, or maybe fibroblasts or neural cells. Some, rat cells are still in these islets. So just in case, we used immunosuppression only for the first 5 days. We're afraid of bystander effect that could damage mouse beta cells. But after all, in all recipients, only 100 islets, but they could normalize the recipient's blood glucose levels from the over 400 milligrams of blood glucose levels. And within 2 months, the GTT returned to normal. And when we removed the grafts by doing heminephrectomy, the blood glucose levels spiked. That means transplanted 100 islets are responsible for this normalization of the blood glucose levels. And most importantly, we didn't do any immunosuppression except for the first 5 days. And because they're immunocompetent, we didn't see any remaining rat cells in the graft. So the host competent immune system must have eliminated all these rat cells present in the initial mouse islets generated in rat environment. So this is the proof of concept data for our ultimate goal, that is exogenic organ can function and can be transplanted without rejection. So we moved onto the larger animals. So we try to do the same for pigs. So we generated apancreatic pigs. That is this one, second from the right. If we know that from our previous experiment, that if we overexpress a transcription factor called HES1, then they cannot form pancreas. So we generated this apancreatic pig. And this is the KUO, means Xavira orange. It's one of those fluorescent gene positive pigs as a sort of marker. So this is the positive control donor pig stem cells. And then these left 3 pigs are chimeric pigs. So then we looked at their pancreas. So this is the apancreatic pig, this one. There's no pancreas. And this is the positive control, Xavira orange transgenic pig. They have pancreas, and they are positive for Xavira orange. And then after we performed process injection using the stem cells from this Xavira orange transgenic pig. Now these apancreatic pigs, they have pancreas like this, and pancreas was very much bright for Xavira orange, indicating that the same idea, process complementation, also works in a large animals like pigs. So this was nice. So we are able to rescue the apancreatic phenotype, and these chimeric pigs grew into adult. So that was very good. So at this point, we're ready to inject human cells. So to do this kind of work, I came to Stanford, the holy land of organ transplantation, to make this realized. So we are still working on this. But during the course of these studies, we realized that it's more difficult to make interspecies chimeras compared to intraspecies, same species chimeras. This is one example with mouse rat study. So these are the same species chimeras. The left is mouse to mouse. Right-hand side is rat to rat. And so about 90% of embryos are chimeric in the case of mouse. In the case of rat, about 50%. Why it's lower in rat? This is most likely because the quality of rat ES cells is not as good as mouse. I think we need to optimize the culture condition for rat ES cells. They're a little different. But anyway, we could get these chimeras relatively easily. And as you can see, these GFP signal more or less, fluorescent signal are more or less homogeneous. But when we tried interspecies chimeras, mouse to rat, rat to mouse, as you can see, the frequency of these chimeras is much lower, about half, 50% for mouse to rat, and maybe 20% with the rat ES cells. And also, most importantly, I think, if you compare these two, you will notice that the fluorescent or staining is somewhat not homogeneous in these interspecies. So there must be some species-specific preference to contribute to the host organs. So we try to understand what's going on here. So we follow we try to see the time course analysis. So this is the days after conception. So up to E9.5, this is rat to mouse chimera. And at this time point, we did see very high donor chimerism, the embryos. But as time goes on, E11.5, E14.5, we lost all these high donor chimerism in mouse embryos. Unfortunately, the organogenesis starts at E10. So by that time, most of these good chimeras are gone. So these also support the idea. So embryos survival rate goes down. This is mouse. More than 80% of embryos survive. So this is not good. It appears that many of the embryos, chimeric embryos with high donor chimerism, they tend to die. Only the low chimeric embryos survive. So this is not quite good for our project. So ideally, it should start low, but eventually goes up. This is the ideal situation for our organogenesis project. So one of my postdocs, Toshia, he found out that IGF-1, which is insulin-like hormone, is important for the organ, the size for the proliferation of cells for bodies, for the whole body and also organs. So, and interestingly, this effect of IGF-1 start to work on the cells after E10 or E10.5, so just about the time to start organogenesis. And when he knocked out this gene, he knocked out a receptor for IGF-1, and it was clearly evident that these knockout mice are much smaller, about 60% reduction in the body size. So if we use these IGF-1 receptor knockout, we thought we may give a sort of competitive niche. So this is not like organ niche. In this case, organs, host organs are not present, but because host cells cannot get this IGF-1 signal because the receptors are knocked out, they cannot proliferate, or they cannot. The wild type cells have an advantage over IGF-1 receptor knockout. So this is a competition between host and donor cells, and only donor cells can respond to IGF-1 and expand. So this could be a interesting system for our organogenesis project, because this could be a multi-organ thing. So we try to see if this really works in the interspecies situation. So this is the rat to IGF-1 receptor knockout mouse embryo. And then, as you can see, we, as you can imagine, the donor chimerism is not as good as mouse, rat. Usually, they have, in all organs, tissues, they have the same level of chimerism in intraspecies chimeras. But in the case of interspecies chimeras, we see a difference in the donor contribution. But interestingly, in some organs like lungs, one of the embryos show very high donor chimerism. So this means more than almost 70% of the cells in the lungs are rat origin. This number is probably close enough to do transplantation. And I think, if we try, the host will take this lung after transplantation. They may need some immunosuppression. But so this system works. And also we tried mouse to IGF-1 receptor null rat embryo. That's the same thing, lots of variations in their chimerism. Organ to organ variation in the chimerism. And in some organs like kidney, they again show very high donor chimerism. So this could be a very promising approach to generate stem cell derived organs. So we generated IGF-1 receptor knockout pigs. And as you can see, this is the wild type. And these are the knockouts. And just like mouse and rats, they show a very similar phenotype, reduction in their body size, about 60%. And unique to this IGF-1 receptor knockout phenotype is their very small thymus and also very small lungs. So using these process complementation, organ-niche approach and cell competitive niche approach, we have generated a number of different organs as shown here. Some of them are mouse, some of them are mouse rat, some of them are also in large animals. And in the case of pig, we have generated double knockouts of PDX1 gene and also FLK1 gene. So these genes are important for pancreas generation, also blood and vessel generation. So in this case, all pancreas, epithelium, and blood and best cells are from injected stem cell derived. So these this should further reduce the possibility of rejection. So we try to move on to human animal chimera generation. So we one of the first trials we did here is to make human sheep chimeras. So this is a sheep embryo, and we injected human marked red iPS cells into this embryo. As you can see here, following 24 hours after injection, we could clearly see human cells still there. Then we transplanted these admixed embryos to sheep foster mother's uterus. But unfortunately, the chimerism after 3 weeks after transplantation, we used digital droplet PCR technology to detect human DNA. And unfortunately, there were some chimeras, but the frequency of human cells in these chimeras was not so high, like 1 out of 8,000 sheep cells. So this was not sufficient to make organ complementation. This is our challenge. So this is not just with sheep, but also we tried human pig and human mouse chimeras and same thing. So this is quite interesting. I mean, somewhat unexpected, because as you know, in all mammals, when we're very young, they all look similar, right? And also, you know that we transplant hematopoietic cells or human neural stem cells to a mouse, immunodeficient mice, but they engraft and grow, no problem. And also, some people at MIT showed that when they transplanted human neural crest cells to E8.5 mouse embryo, some of them survived and engrafted. So mice can take human cells if they are later stage. But from our study and also others, most human cells are eliminated before E6.5. So we just cannot keep them up to this stage of mouse development. So this has been a challenge to make human-animal interspecies chimeras. But we have made some, of course, progress. And by introducing some genes that can make human cells resistant to apoptosis, and also to give some proliferative activity, extra proliferative activity, then these days we can see human cells still present and well integrated with mouse cells. For example, here you can see human cells very well integrated with mouse tissues or cells. And this is vessels. The GFP signal is the human cells, and this is the mouse, mouse-human vessels, very well integrated, and they must have differentiated in a tissue. And these are not fusion of these two genes. We have genetic evidence that they are the human cells expressing vessel-specific or cardiac muscle-specific genes. So by just doing some genetic modification, we are able to keep those human cells stay alive and integrate, co-develop with mouse cells. I hope this will work better with a human-pig situation. So far, we found that there's a very tough, strong xeno barrier, a very early stage of embryogenesis. And most, as I mentioned, most human cells are eliminated if there's no genetic intervention. So this happened before organ niche or cell competitiveness start around E10. So we have to, somehow get over this xeno barrier to make human animal chimeras or make human organs in animals. But we thought that is there a way to create organs going through this early phase xeno barrier? What about complementation of organs at later stage, like at this stage, via in utero transplantation? So we thought about this and we tried to target skin. So skin is made of epidermis and dermis, and there are some commercially available products for the keratinocytes, cultured epidermis. But these products, they don't have, they're very thin, and they cannot engraft because they don't have dermis, and no hair follicles or sweat glands and so on. So we thought we may be able to generate transplantable skin using in vivo environment. So we tried this first with rodents, a mouse system. So we know that P63 is essential for the differentiation of keratinocytes. So we generated P63 knockout mice and lemurs because skin is necessary for the development of limbs. So these are skinless, limbless embryos. But when we, of course, performed racist complementation, these defects are sort of, rescued. So they have limbs and also they have skin. Some part is still missing some skin. It takes more time, I guess. So this worked nicely. But the point is, this is just a racist complementation between mouse, so it should work. But with a human, it should be difficult because they have to get over this xeno barrier in early stage. So instead, we thought we may be able to generate human skin because this niche is outside the body of the embryos. So we thought if we inject skin progenitor cells, keratinocyte progenitor cells in the amniotic space. Anyway, this space by in utero injection. Technically a little difficult with a mouse, but it's possible. And we try to see if this works. These P63 knockout embryo provide some niche for the skin development. So we did this experiment, and indeed, one of my students injected human keratinocyte into this amniotic space and succeeded in making this human skin flap. It's still small because it's a mouse, and it only had 6 or 5 days before they're born, so not much time to expand. But still, we were able to see human cells when injected as a single cell suspension. They still stick to the body of the embryo and form the keratinocyte layers. So this is interesting, I guess. So we're hoping that if we use P63 knockout pig embryos, then it should be a little easier to inject human keratinocyte or skin progenitor cells. Could be from the patient or could be from the iPS cells. And then because pig has much longer gestation period, like over 114 days, so they should have sufficient time to expand and make nice human skin flap. So this project is now going on. So overall, I mentioned we found that very tough xeno barrier in a very early stage of animal development, and without get over this xeno barrier, it's difficult to perform organ complementation using process complementation or IGF-1 receptor knockout. But in utero is another potential system to generate human organs. So we are now trying to introduce various genes to human iPS cells to make them stronger, more resistant to death, or they can make a better integration with mouse or host animal cells. So we are working on this on the human iPS cell side. And the host animal side, we generated organ niche, also cell competitive niche. So by doing these trials, experiments, I hope we can generate human organs in vivo soon. So I think it's important to understand and manipulate this xenobarrier. That will be the key to the generation of human organs in livestock animals. So these are the names of the contributors and funding agencies. Unfortunately, NIH still does not support human-animal chimera research. So I stop here. Be happy to take any question. Thank you for your attention.